So listen, I've been reviewing some of the many problems with the theory of evolution, and now I want to focus on two, not unequally. I told you that David Gelernter, who's a professor of computer science at Yale, wrote a review of one of Stephen Meyer's books in the Claremont Review of Books, which is a review of academic literature, and he called it Farewell to Darwin or Goodbye to Darwin, something like that. He explained why, from having believed in Darwin's theory, he changed his mind. And he mentions a number of problems, one of which is a famous problem that's still continuously debated, and he gives a summary, and then and then one of them is a calculation, which I hope I can explain to you reasonably clearly, which is not a question of it counts against, and who knows how much it counts against, and maybe there'll be an answer, and so forth and so on, but a direct calculation of impossibility. So let's take a look at the first one. Something called the Cambrian explosion, about 500 million years ago, and that's the term that's used in the standard evolutionary literature, the Cambrian explosion. A striking variety of new organisms, including the first ever animals, pop up suddenly in the fossil record over a mere 70-odd million years. And I say, 70 million years? That's a long time, Rabbi. What do you mean mere? Yeah, but not when you're talking about the span of life on this planet. That's 4.3 billion years. 70 million is just a blink of an eye, especially, as you will see, for brand new body plans and animal organization to pop into the record over such a period of time. This great outburst followed many hundreds of millions of years of slow growth and scanty fossils, many of single-celled organisms, dating back to the origins of life roughly 3.5 billion years ago. Darwin's theory predicts that new life forms evolve gradually from old ones in a constant, branching, spreading tree of life, which we spoke about yesterday. These brave new Cambrian creatures must therefore have had Precambrian predecessors, similar but not quite as fancy and sophisticated. They could not have all blown out suddenly like a bunch of geysers. Each must have had closely related predecessor, which must have had its own predecessors. Darwinian evolution is a gradual, step-by-step process. All those predecessors must have come together further back into a series of branches, leading down to the long ago trunk of the tree of life. That's the Darwinian picture. But all those predecessors of the Cambrian creatures are missing. They're all missing. You go from stuff like sponges and and algae to animals, complex animals. In fact, if I remember correctly, 12 different body types of animals in that short period of time. All the predecessors are missing. Darwin himself was disturbed by their absence from the fossil record. This was known 150 years ago. It wasn't something they discovered yesterday. He believed they would turn up eventually. As I told you yesterday, the name was Stephen Stanley. He thought the fossil record is incomplete, and we haven't found so many fossils. We dig so well, we'll find them. He believed they would turn up. Some of his contemporaries held that the fossil record was clear enough already, even 150 years ago, to say Darwin's theory's got to be wrong. Perhaps only a few sites have been searched for fossils, but they have been searched straight down. Some places they went straight down through the Cambrian period and to before and found nothing. They thought it was already bad in 1869. The Cambrian explosion had been unearthed. You could see that in going straight down, that there is this sudden appearance of all these body types. And beneath those Cambrian creatures, their Precambrian predecessors should have been waiting and weren't. So even in 1869, people said it was bad enough. In fact, the fossil record as a whole lacked the upward branching structure that Darwin predicted, which I told you yesterday. This is now this business of being static and then just jumping. Some researchers have guessed that those missing Precambrian precursors were too small or too soft-bodied to have made good fossils. Whether a dead creature survives, its body survives long enough to fossilize. If it's very small, maybe it'll dissolve, or maybe it'll be crushed by forces. And if it's soft-bodied, then the bones have their own structure, and you can fill them in with stone gradually. But the soft body will just deteriorate. Maybe that's it. Now, that was what some people thought. Stephen Meyer, the author of the book, notes that fossil traces of ancient bacteria, which are one-celled creatures, and single-celled algae have been discovered. Smallness per se doesn't mean that an organism can't leave fossil traces. So don't tell me they're too small, because we have them single-celled. That's as small as you get. Although the existence of fossils depends upon the surroundings and the history of the rock, that's true. The story is similar for soft-bodied organisms. Hard-bodied forms are more likely to be fossilized. It's true. Bones are more likely to become fossils. But many fossils of soft-bodied organisms and body parts do exist. So don't tell me they were too soft to be fossilized. That's not true in the empirical record. So those two excuses don't work. Precambrian fossil deposits have been discovered in which tiny, soft-bodied embryo sponges are preserved. So Precambrian, you have preservation in the fossil record of life forms, but not precursors to the Cambrian explosion. No precursors to the celebrity organisms of the Cambrian explosion. Now, Gerlanter is a careful logician. This sort of negative evidence can't ever be conclusive because there are too many factors involved, and maybe we unfortunately looked in the wrong places, or maybe we'll discover some new explanations as to why they aren't there. But the ever-expanding fossil archives don't look good for Darwin, who made clear and concrete predictions that have so far been falsified by his own admission. Because Darwin says that the fossil record in his day doesn't support his theory, and it hasn't gotten better. So nobody's throwing a brick at Darwin. Darwin, in his honesty, is admitting that if the fossil record remains like this, it's bad for my theory. But bad for isn't yet a refutation. It's just a problem. When does the clock run out on these predictions? No. And if another 100 years, another 200 years, we don't find them, should we then say, time's up, must be wrong? Maybe never. But a thoughtful person must ask himself whether scientists today are looking for evidence that bears on Darwin. Are they neutrally and objectively looking, both positively and negatively, to find evidence that's relevant to Darwin? And objectively and neutrally noting its relevance, in which case they would have to say, yeah, Cambrian explosion counts against Darwin? Or looking to explain away any evidence that contradicts him and not taking it seriously because they're committed to explaining it away? Are they looking at it objectively or in a partisan fashion? Read your exposition of evolution and see how they treat the Cambrian explosion. Do they admit that it's a problem? Do they wave it away? It's not a problem, it's easy to explain? Well, Darwin didn't think it was easy. Why do they think it's easy? There are some of each. There are some scientists who are doing it objectively, and some are doing it in a partisan fashion. Scientists are only human, and their thinking, like everyone else's, is colored by emotion. So if you want to look these things up, look up the Cambrian explosion. However, Darwin's main problem is molecular biology. There was no such thing in his own time. They couldn't see molecules. They couldn't even calculate them. We now see from inside what he could only see from the outside, as if he had developed the theory of mobile phone evolution without knowing that there were computers and software inside the digital revolution, what it's all about. Under the circumstances, he did brilliantly. Biology in his time was for naturalists. Okay, that is. And then he describes Darwin's theory, which I described to you in exactly the same terms. As I said yesterday, I said it as an aside. You have sheep, and there are natural variations in the wool on the sheep. And if you have a mutation which causes the wool to be thicker and therefore warmer, then that sheep is going to survive cold better than other sheep is going to survive cold. And other things being equal, they'll have more offspring. And then they'll improve the stock of sheep and so forth and so on. That was the kind of observation that was used from the outside without knowing anything about molecules. The advent of molecular biology made it possible to transfer Darwinism into neo-Darwinism. That's their own term of the Darwinist believers. The new version doesn't merely cite natural variation as the consequence of radium change or mutation within cells. Sorry, I didn't read that right. The new version explains natural variation as the consequence of random change or mutation to the genetic information within the cells that deal with reproduction. We don't just say that things change, look and see that they're not all the sheep are the same. We have the DNA, and we explain that the changes that you see in what's called the phenotype, the presentation of the whole organism, depends upon the DNA. We know how it works. Of course, that's what Darwin says. These cells, they're Darwinians, these cells can pass through genetic change on with the next generation, thus changing, accidentally, potentially, the future of the species, not just one individual's career. The engine that powers non-Darwinian evolution is pure chance and lots of time. You must have heard this at least 100 times. But it's billions of years. Anything can happen in billions of years. Let's see. By filling in the details of cellular life, molecular biology makes it possible to estimate the power of that single mechanism. But what does generating new forms of life entail? How must it be done? What's the mechanism by which it happens? Many biologists agree that generating a new shape of protein is the essence of it. Only if neo-Darwinian evolution is creative enough to do that is it capable of creating new life forms and pushing evolution forward. Now, why are proteins so important? He explains. Proteins are the special ops forces, or maybe the Marines, of living cells, except that they are common instead of rare. They do all the heavy lifting, all the tricky and critical assignments in a dazzling range of roles. Proteins called enzymes catalyze all sorts of reactions and drive cellular metabolism. You've heard of enzymes, they're proteins. Other proteins, such as collagen, give cells shape and structure. Nerve function, muscle function, photosynthesis, all are driven by proteins. And in doing these jobs and many others, the actual 3D shape of the protein molecule is important. Now, you have to understand, himself is going to explain, a protein is a long chain of atoms run together. Organic molecules can create chains of almost unlimited length, but if it's stretched out as a chain, it does nothing. Some proteins automatically bind up into complicated folded shapes. Some of them roll, some of them jackknife and jackknife and jackknife again. After the jackknife, they twist into a twisted shape. After that, they tie up in knots. They do all sorts of automatic things. The folding of proteins is an entirely unsolved problem. They think that maybe with quantum computing they can solve it because they're given the number of atoms, which, as you'll see, is large, and given the number of particles and all the electrical forces between them, to predict which way a protein is going to fold is a gigantically difficult computational task. Some fold, some don't, and some actually fold into more than one shape. They can switch the shapes. They can switch the shapes on demand. The functionality of the protein depends upon the folded shape. And just to give you a little hint of why this is so, DNA is not a protein, But DNA, the DNA in your cell is about, if I remember correctly, 6 feet long. 6 feet long, and it fits inside the cell. How does it do that? Because it rolls up and folds up and twists up and contorts itself. Okay, now imagine it's rolled up into a ball. Now think about it. This ball is inside the cell, surrounded by a fluid and surrounded by all sorts of working machines, but not all of the DNA is equally available to the fluid. Because some of it's inside, buried inside thousands of layers. So it's harder to get an interaction with the inside piece than it is to get an interaction with the outside piece. And that's going to determine how the functionality of the DNA operates, right? Yeah, but the DNA changes its shape. Changes its shape. So different parts can be operated at different times. Isn't that clever? All by accident, of course. Right? So the protein's the same thing. When you fold it up, some parts of the protein are available to immediate interaction. Others, to interact, you'd have to crawl in between the spaces and get inside. The shape of the protein is absolutely crucial to its functioning. This is well known. This isn't speculative. So now, if we need all those proteins, we first have to ask, what kind of molecule is a protein made out of? And then we have to ask, how does it bend? How does it fold? That's going to be the key to the argument. So first of all, he explains how mutations work, and but I'll read what he says because he's got other stuff here that's important. Proteins are chains, linear sequences of atom groups. So each thing in the chain, you think of beads on a string, each bead is not an atom, it's an atom group. A protein molecule is based on a chain of amino acids. So each bead on the chain is an amino acid, which itself is a complicated molecule, but may have 35 atoms. 150 elements is a modest-sized chain. The average size is 250. So if we talk about 150 as the model for the barrier for evolution, we're making a very modest assumption. We're taking ones well below the average. So if we ask whether evolution can do this, and it can't, then it's sunk, because it can't even do easy ones. So now you got a chain with 150 beads. Each link, each one of the beads, is chosen ordinarily from one of 20 amino acids. Aha. Imagine a kid who's making the chain. He's making a necklace. So he takes the piece of string, and he looks down. He's got in his box 20 different little sacks of beads. Blue ones, yellow ones, magenta ones, and so on. And he picks one out of 20. 1 out of 20 for each of 150 spots. How many necklaces can he make? Lots and lots? Yeah, lots and lots. It's the number 20 to the power of 150. 20 to 150. That's how many chains he can make. Now the question is, out of all those chains, what proportion are well formed? What proportion are going to fold up into useful shapes? Okay? So then you know that if there's a certain total of all possible shapes, and you have a percentage of good ones that will work, and you're working with a random process, you can make a very simple calculation. What's the probability on a single try of getting one of the ones that works? And then you can ask, in the whole history of the universe, or the whole history of life, how many tries have there been? Now let's say that there were 200 possible chains, and the probability of getting a good one is 1 in 1,000. But suppose there have been 1,000,000 tries. With 1,000,000 tries, we'll get them all the time. It all depends upon how many total possibilities there are, what percentage of them are good, and how many tries have been made. That's how you make the calculation. Now, we have to just do one more level. How are we getting these proteins? We're not taking amino acids and shaking them up in a bag and hoping they stick together. Amino acids are built by DNA. That's how it's done in cells. We're not talking about origin of life now. We're talking about how life develops and continues. So you've got cells, you've got the DNA, and the DNA directs creating molecules. The DNA is like an instruction kit. The RNA copies it and brings it to a machine, and the machine builds the protein or the molecule that the instructions tell you to build. So for the proteins in the cell, there's a stretch of DNA, each one for the protein, which tells the machine what molecule to build, in particular what proteins to build. You want to get new proteins, you got to change the DNA. This is where mutations come in. Okay, so now, and each link in the beads, each bead on the necklace, the amino acid has one piece of instruction in the DNA specifying that bead. So if the thing is 150 beads long, you got 150 instructions in the DNA telling you, put this bead here, put this bead here, put this bead here, put this bead here. To get a new one, you got to change one of those instructions. Then to get a new amino acid in that spot, to get a new necklace. At least new in one place. So now, you want to know how you get a new protein. Now, so you've got your DNA, and in it there are stretches that give the instructions for each protein, and other things as well, that are built in the cell. That's not all the DNA. According to Orthodox Darwinism, I think this is mistaken in many ways. I think this is mistaken in many ways, but all right. We're testing Orthodox Darwinism now. I want to turn this off. Yeah. So the DNA has some instructions for building proteins, and it has a lot of gibberish. A lot of the DNA doesn't do anything. It's just random junk. That's what they say. It's called junk DNA. So now the biologist asks, if we're thinking about creating instructions for a new protein, and we're going to do it by mutation, where should we look? Should we look at the instructions for an old protein and talk about mutations in that instructions to an old protein, which are going to then make it into instructions to a new protein? Or should we look at the gibberish? Well, the description of the protein is so complex that if you take the description of an old one and change it in any way, it's going to fail. But it's not going to be gibberish. The idea of starting with gibberish and working it step by step into a new instruction is much easier than doing it with something which is already dedicated to an old instruction, which you'd have to take apart completely and then start over again. So they're starting with this, the favorite side. Start with gibberish, talk about random mutations, and see how many random mutations you would need to create out of gibberish an instruction for a functioning protein. So, this is the calculation that they made, as he calls it, building a better protein. He says, now we're at last ready to take Darwin out for a test drive. Starting with 150 links of gibberish, it's 150 for each one for the one that's of the pieces of the protein that's going to be built. What are the chances that we can mutate our way to a useful new shape or protein? We can ask basically the same question in a more manageable way. What are the chances that a random 150 link sequence will create such a protein? Nonsense sequences are essentially random. Mutations are random. Make random changes to a random sequence, and you get another random sequence usually. So close your eyes, make 150 random choices from your 20 bead boxes, string up your beads in the order in which you chose them. What are the odds you'll come up with a useful new protein? It's easy to see that the total number of possible sequences is immense. It's easy to believe that the subset of useful sequences is in comparison tiny, but we must know how immense and how tiny. Remember, there are billions of years. That's the mantra of the believers. Anything can happen in billions of years. Well, let's see. The total number of possible changes is 20 to the 150, which I told you. Now let's just put some numbers on the board so we have them to deal with. $20 to 150, which is about, because we're going to compare it with others, about 10 to 195. You know what that symbol means? That means a 1 with 195 zeros after it. That's a lot. That's a real lot. Okay? What proportion of these is useful? Now, a molecular biologist did the following calculation. What proportion of these will fold? Folding isn't enough. Not every folded shape is useful. But what proportion folds? We're giving an extra advantage to the argument. We're not asking for the ones that are really useful. We're asking for a much bigger set, the ones that will really fold. Stable folds. Okay? The proportion is, this is stable folds, is 1 out of 10 to the 74. 1 out of 10 to the 74. So what we're really looking for is, what's the probability of getting one of these? 1 of 10 to the 74. That's a 1 with 74 zeros. Well, 1 out of 10 to 74 is 0. If you have to, win it at poker with 1 out of 10 to 74, you can give up. Okay? Okay, now he says that's stable folds, but functional stable folds is less, because not everything that folds is useful. And the estimate to that is you need to add 3 more zeros. So it's 10 to 1 to the 77. So if you're looking for the functional proteins, your chance of getting it by random process, and Darwin's process is a random process, is 1 in 10 to the 77. Okay, but now, in other words, the immense is so big and the tiny is so small that neo-Darwinism is so far a dead loss. But we have to understand, we're not doing it once. If you did it once and the chance of it 1 in 10 to the 77, you just give up. We're not doing it once. How many chances are there for mutations? If you talk about bacteria, there are trillions and trillions, maybe quadrillions and quintillions of bacteria. And they've been around for billions of years. So you got to know how many chances are there to do mutations and see whether maybe you can get up to 10 to the 77. Maybe you can make it probable. After all, there are billions of years, as the mantra, right? Are you taking that into account? Well, let's try. So, he says, the guy named Douglas Axe says, first of all, let's again be generous to the argument and say that every bacterium that has ever lived has had a mutation. Every last one of them. That's surely not true, but we're giving it the best chance, okay? How many bacteria have there been? Well, the estimate is about bacteria in the history of life, 10 to the 40. That's a lot. So that means you get 10 to the 40 mutations, being overwhelmingly extra generous beyond what life would actually support. Well, if you take 10 to the 40 tries to get something that's 1 in 10 to 77, you end up with a probability of 1 in 10 to the 37. That's not going to do it. That's a 1 with 37 zeros. That's the probability of getting life through the Darwinian process. Not life, just proteins. Just proteins. There are 1,000 other things you need. This is a tiny sliver of evolution with a calculation in specific enough terms is that there's nothing left to talk about. Yeah.
Is there a word for a number that big, or do you just have to say 10 to the power of 37?
10 to the power of 37.
Wow.
Yeah. So now, I've asked people about this, and the literature that I've seen on it is to call him names and say he's a creationist, and he's against modern science, and so forth and so on. But I haven't seen anybody say, no, it's not really bacteria, it's also snails. Or, you didn't subtract correctly. 73 and 4 is not 77, it's something else. Or, where exactly in the argument is there some kind of mathematical or scientific failure here? You cheated because you made it too difficult, and so forth and so on. It'll be interesting to hear. But so far, as I told you, he's a professor of computer science at Yale, so he's not dumb. Yeah.
How did he formulate that probability or those figures with respect to that probability? Where did he get those numbers from?
He didn't get them. He's reporting on a book, which was this was done research by molecular biologists.
And it's noted there?
Yeah, of course, all the footnotes are there.
So it's saying you have the 1 in 10 to the 37 chance, but how many of these proteins are being made? If you're making 10 to the 500 proteins a day, right? This kind of could make sense, right?
Oh, sure. Scientists do this in laboratory all the time. They make proteins all the time because you have an intelligent design way to make proteins, to manipulate proteins. We're questioning whether it happened through the Darwin process. We're not saying it's impossible to make proteins. The question is whether the Darwin process could be expected to make proteins. And as I said at the outset, when you tell me something happens by a non-directed process, everything hangs on the probability. Now, I hope I'm not causing a confusion here. I said you can't have any probability for the whole process yielding the whole envelope of life. That's what was going on here. We're taking an extremely narrow 1,000th of the story, defining it very carefully, talking about a specific process and asking what is probability. That you can do. And this may sound prejudiced, but this is the fact. You can use probability to defeat, but you can't use probability to support, because they're different probabilities. If I find one tiny sliver of the story which probability says can't happen, the game is over, because the story depends upon that sliver. If I can't find anything that defeats any sliver, still the question will be, why should I accept the whole story when I can't get a probability of the whole story? Those are two entirely separate, independent considerations. This is a tiny sliver of the story according to the way in which they say it should happen, and the probability is so overwhelmingly immense that it just doesn't allow for it. You got a question? Okay. Yeah.
So what would a Stephen Jay Gould or a Richard Dawkins say about that?
Google it and try to find out. Google it.
Well, one of them's dead, I think, right?
Who?
Isn't Gould dead?
Oh, sure, but so what? He has plenty of successors. Yeah. There are plenty of believers. Jerry Coyne is a real, true believer. Nothing phases him. Nothing counts. Nothing matters. If you say it, you're lying, or you're prejudiced, or you're incompetent, and that's the way the people. That's the way they argue. They argue, and they poke fun at people, and they make fun of their politics, and for which I told you on another occasion that should make you suspicious as to whether or not you should give them any credibility. They should just answer the argument straight out. So just Google it and see if you can find someone who points to the specific problem. Your argument goes from step 16 to step 17. That's wrong, because 16 doesn't imply 17, and here's the reason. See if you can find it. I can find it. So, and this is published in the Claremont Review of Books is a very famous review of academic books, written by academics themselves, not as if it's hidden away in some obscure publication. So this is a positive reason to say that evolution is false. Something I haven't given you up until now. Up until now, I told you I was arguing against the credibility of the support for evolution. I told you if I was successful in the previous stages, we should admit simply that we're ignorant, we don't know whether it's true or not. This is a direct argument against it. Now, this sort of thing has been going on for 40 years. The Wistar Conference, I think it was the 60s, of mathematicians who tried to apply mathematics to Darwin, and they said they came up with impossibilities. But they were applying to things which weren't precisely defined. They didn't have a molecular picture. So they were talking about features and time and general circumstances, and they tried to make a mathematical model, and none of them worked. But that could have been a failure of the information that they had. This is much better defined. We have molecules. We study them. We can look at them under electron microscopes. We know what they're made of. We know what their forces are that operate on them. And we know the circumstances, and we know the kinds of mutations that take place. And this argument is very robust. We're not saying when there are cosmic rays, so it's more likely to have mutations than when not. This argument overwhelms that. If you assume that every bacterium in the history of life had a mutation, that's surely vastly, vastly more than the mutations that took place. If that's not good enough, then the real world is surely not good enough. So I think that's something to keep in mind when you're talking about evolution. Questions up to here? Okay, so yeah. Sorry. So, yeah.
What do you mean? I'm not really familiar with like sciencey stuff. I'm just wondering what you mean by like stable folds and.
Okay, let's see if I can give you an example of this. Let's take the folding of a bird's wing.
Yeah.
It's really complicated. Now, it would be exhausting for the bird to have its wings extended all the time and to have to walk that way and eat insects off the ground that way and so forth. It's extremely useful for the wing to fold and fold in such a way that you have a streamlined body shape. Right? So imagine you have these flaps which are very useful, but they don't fold. That's going to be very difficult. And now you have to engineer new genes which have new muscles, and new muscles have to be coordinated. These pull first, and those pull next, and then some things to hold it in place, and you're doing it at random. It's a complicated system, similar to what I said about the giraffe, where you have to have more skin, more vertebrae, longer neurons, you have to have a stronger heart, blood vessels carrying the blood to the brain, and not gravity sloshing it back down, so you have hinges in the blood vessels which stop and hold the blood from sloshing down. The balance organ in the head has to be reprogrammed so as to be able to balance an organism where it's 6 feet above the main body, which it isn't in the thing that it's evolving from. All these changes have to be coordinated. They all have to go, and they have to go gradually. Because if you get the growth of the head without the stronger heart, it'll die because the blood doesn't get to the head. All of them have to be coordinated. This is very, very complicated. For a protein, first of all, the beads in the chain have to be strung together in the right order, some order that holds. But then some of them naturally fold up. Why would they naturally fold up? Because let's say the bead at this end organizes its atoms in such a way that the electrons are prominent as a negative charge. And the bead on this end organizes atoms so that the electrons aren't prominent, so the protons have more of it. So then you got the proton, the positive magnetic fraction here, and negative here. So the they're going to fold together. Something like that. But you got 150 beads. And it's got to fold up into one shape that holds stably. Not every protein is going to do it. Not every string of beads is going to do that. So knowing what the forces are that hold them together, what counts as stability, they calculated this proportion would be stable. Yeah.
What do you make of epigenetics? Like, for example, the fact that certain Jewish people went to different climates and became like darker skin and darker hair, or like, you know, certain like activities will like change your own body, and then that'll get into your gene pool, and your kids will have those qualities.
Well, I'm not informed. You may be better informed than I am. I'm not informed that, let's say, skin color is due to epigenetics. Epigenetics means there are features in the cell which are not the DNA, and they contribute to the functioning of DNA, and therefore they can control how and when and in what way the DNA functions without changing the DNA, which means there's more flexibility in changing the phenotype, that is the way the body looks, than just mutating the DNA, which gives much more flexibility in adaptation. Now, there are, I forget what they're called, there are certain types of molecules that can attach to the DNA and simply silence it. So it's not functioning. After all, every one of the cells in your body has the same DNA. They're very, very different from one another. A nerve cell and a muscle cell and a red blood cell and a skin cell look different, different sizes, different shapes, and different functions. How do they do that? They all have the same DNA. Answer is, not all the DNA is functioning all the time. Guess what, men? It was discovered, maybe it's only 2 years old, that the DNA that a male passes to the female in conception may carry features of his life experience. The idea of Lamarckian evolution isn't dead. And here's how it happens. Because the formation of the male zygote, when it goes from the testes to the exit, it takes about 2 weeks. There's a long, complicated tube that it goes through before it can be expelled. And during that time, the DNA is treated by other features of the body, which, due to its experiences, favor the expression of some parts of DNA and disfavor others. And that DNA in the sperm is now loaded with information of what should function and what shouldn't function. And that is passed on to the female with that loading in it. So it isn't just pure DNA, it's loaded DNA. It's pre-sifted DNA. So for example, and they found this in people, there was a population somewhere in Europe which went through a terrible famine, and the body developed resistance to the famine, ways to deal with the conditions of famine, and 3 or 4 generations later, they found people, the descendants, who didn't live under famine, had the same features. Which means the experience of the famine caused the body to respond in a certain way, and that response was passed down to later generations. We know this with single cells. That's been known for a long time. You attack a single cell, and with its DNA, it has a certain defense. And when it splits into 2, it's carried along. When it splits into 4, it's carried along. So but again, this is not mutations. You're not changing the DNA. You're changing the way the DNA functions. So once you do that, so that means a man can pass on some of the experience of his life to his children through his DNA because of the sifting of the functioning of the DNA in the sperm before it's expelled. I mean, these things are, the system is much more complex, much more subtle than anybody ever expected. And I want to tell you one more example. I've got to tell you this. This is just beyond belief when you talk about things that happen by accident. So I told you that you have the DNA, and then it gives instructions to the machines to make proteins, right? I told you this, according to the official story, there's junk. Junk DNA, stuff that doesn't code for anything. So you sort of picture, here's a stretch that's instructions, and there's a stretch of junk, and here's a stretch of instructions. It doesn't work like that. Because when you look at the stretch of instructions, in between the instructions there's junk. They're called introns instead of extrons. Introns. So it's a stretch which contains instructions and junk. Wow. So it's got junk in there. What do you do? Well, here's what happens. The RNA copies it. It's got the whole same structure. Then the RNA breaks into pieces. The parts that are junk drop out, and the rest of the parts are sewn together, and now you have only instructions. And now the instructions go to the machine. Tell the machine what to build. What a kludge! What a stupid, silly, inefficient system. Except they discovered that when the RNA copies everything, including the junk, and then breaks up, and the junk drops out, the remaining pieces can be put together in more than one way. The remaining pieces can be put together in 20 different ways. And each way of putting it together programs for a different product in the machines. One stretch programs 20 products, not one but 20. And guess what? The junk that drag out is the stuff that stuffs it together, strings it together. Not a dumb system, hey? Not as kludge, not a stupid, silly system. The contrary, you show me your computer program that can do that. One stretch can do 20 different jobs? Sorry, ain't the kazeh. The system is much more complex and sophisticated than anybody dreamt, and they're discovering new things about it all the time. You know, I told you, and you know about the double helix unwinding, and you get one each, and then you split, and then you make copies, right? You're making copies. How do you make copies? Well, there are these machines, and they bring in the right thing. That they always get it right. There are molecules which are checking the construction, and if it goes wrong, it stops it and corrects the mistake. And every week they're discovering new stuff. Every week they're discovering new stuff. Sometimes, like I told you about the DNA, the functioning requires one piece of DNA to be close to another piece of DNA. So it depends on how it's folded. That's one of the reasons the folds change. From time to time, based on the circumstances in the cell, it comes time for them to interact, so it shifts its shape, and then this part is close to this one, and they work together, and then it shifts again, and it's not together again. So, and of course it all happened by chance, yeah.
So what you're talking about, the experience of a man being transmitted through his semen, and then children get it. If a large enough population got this thing, it would change the whole population. Isn't that sort of like evolution?
Well, the question is whether it continues forever. This is not mutations. Let's be careful. The DNA stays the same DNA. There's certain types of atoms that can attach to the DNA and mute it so that these parts of the DNA don't interact with something else. Just as they can attach, they can detach. There is some thought of actually changing the DNA. That's something newer, which I have seen some references to. I don't know how well it's established. Even the mutations can be influenced by the circumstances. That's a gigantic revolution. Because they calculated the probability of a mutation, they found in certain circumstances the number of mutations goes up, and other circumstances it goes down. That sounds like the circumstances having an effect on the mutations. That would be like absorbing programming from the environment. Weismann's law said that information flows in one direction only, from the DNA to the RNA, from the RNA to the machines, from the machines out. There's no influx of information to the DNA because Darwin said all the variation is random. But they may not be true. That may not be true. Okay, there's endless stuff here. Last question, and then we'll continue it tomorrow.
I don't know if you had any thoughts on, like, when you look at the DNA of a human compared to, like,
Oh, yes. The chimpanzee is 98.6% human, hey? We'll talk about that.